So this is the introductory video to the Endocrine System lab in the Cell Structure and Function course. The goals of this short video are to help you understand the structure and to begin to understand the functions of the pituitary, thyroid, and adrenal glands. The first slide is a very low magnification image of the entire pituitary section of the pituitary, which was from a cadaver. As you can see from the labeling, there are several parts to the pituitary. The portion labeled D is the pars distalis, which is a Latin phrase meaning the distal part. It's also known as the anterior pituitary. And, just to make your life more difficult, it's also known as the adenohypophysis. So 3 different names mean exactly the same thing. To the left, the structure labeled N is the neurohypophysis, or the pars nervosa. It's also known as the posterior pituitary. The portion labeled S is known as the stalk, or the infundibular stem, and the region labeled T, which here appears as a very thin structure, is called the pars tuberalis, and pars tuberalis is related to the pars distalis, and the infundibular stem is related excuse me, to the pars nervosa. In the middle is a structure here that we call the pars intermedia, and at this point we don't really understand the function of this particular portion of the pituitary. What we're going to do now is to look at a region of the pars nervosa at slightly higher magnification. This slide shows you a region of the pars nervosa. We're still at low magnification. It's very difficult to make out nuclei here. Indeed, this portion of the pituitary is almost exclusively made up of axons. The cell bodies for these axons are in the hypothalamus, and you don't see those in this preparation. You only see the axons that have descended through the infundibular stalk down into the pars nervosa. So indeed, as you recall from the nervous system lab, the structure here is difficult to make out, basically because you have lots of cell processes and relatively few cells that are present. In the next slide, which is now a medium magnification, we can begin to see a little bit of structure in the pars nervosa. These spaces, or lumens, are blood vessels, capillaries, that run in the pars nervosa. And within the axons are accumulations of neurosecretory substance, and they have been given the name herring bodies. That's why they are labeled here with an H. And the herring bodies contain, as you will learn in lecture, 2 substances. One is called oxytocin, and the other is called antidiuretic hormone, or vasopressin. There are some cells here in addition to the endothelial cells, and these cells are called pituocytes because they are supportive cells within the pituitary. They are difficult to make out in this sort of a micrograph. You would only expect to be able to see some very small nuclei that are associated with the axons in the pars nervosa. So now we're again looking at the entire pituitary in cross-section. And here's the infundibular stalk coming down into the pars nervosa, which is what we just looked at. As you learned about in lecture, the pars tuberalis carries blood vessels with releasing factors down into the pars distalis. So now what we're going to do is to look at a region of the pars distalis in higher magnification. For example, the area that is within the circle. So now we're looking at a high micrograph, excuse me, a high magnification light micrograph, because you can now easily make out nuclei and even small nucleoli within the nuclei, and you can see the cytoplasm surrounding the cells. You'll note that these cells are arranged in clusters, or what we call cords. They are epithelial-like, and they sit along the basement membrane that they create, and they sit right adjacent to capillaries. These are fenestrated capillaries, because these cells are secreting hormones that then go into the blood and are moved out to other parts of the body. You can see differences based upon histological staining. Excuse me. We see here this reddish staining cell, which is called an acidophil, which is red or pink in color. And then we also have a cell that stains blue. These are called basophils because they attract a bluish stain in this preparation. And then there are cells that don't stain with either red or blue. They have a cytoplasm that's clear in appearance, and these are called chromophobes, implying that they do not take up stain. And these staining differences indicate for the acidophils and the basophils, these are the cells that contain the different hormones, the 6 different hormones that are secreted by the adenohypophysis. 6 hormones that you will learn about, 2 of them secreted by the acidophils, are growth hormone and prolactin. So those are the red staining cells. The blue staining cells secrete thyroid stimulating hormone, follicle stimulating hormone, luteinizing hormone, and adrenocorticotropic hormone, or ACTH. We won't ask you to memorize which hormones are secreted by the acidophils and basophils because we can use very different stains to look at this particular organ, but you will be required to understand the different hormones that are secreted by the pituitary, both the anterior and posterior pituitary, and the targets of those hormones and how they regulate metabolism in the body. And you will integrate this information with Dr. Burdick's lectures in biochemistry as he tells you about cell signaling and how hormones interact with receptors to affect changes that alter what goes on in normal body function. So now we'll move on to the thyroid gland. This is a very low magnification of the thyroid gland, and I'm tracing with the arrow the outer extent of the thyroid gland that is present in this image. This structure, which is immediately adjacent to and actually embedded in the thyroid gland, is called the parathyroid gland, and this is a gland that is responsible for calcium regulation. The thyroid gland secretes the thyroid hormones T3 and T4, which have a wide variety of effects on body metabolism. At higher magnification, the thyroid gland has the appearance of what we call follicles. And follicles are roughly sphere-shaped and cut in cross-section. You can see a cuboidal epithelium that surrounds a lumen, and within that lumen is a substance which is the precursor of T3 and T4. It is given the generic name of colloid, which really is not specific in any sense. It's a very general descriptor. But please understand that this is material that is synthesized by the epithelium of the thyroid gland and stored within the lumen. And here we see many follicles, 1, 2, 3, 4, 5, 6, 7 follicles occur in this image. Just at the base of the epithelium, you can see endothelial cells of the vasculature. This is a highly vascular organ, as are all endocrine organs, because these organs are secreting hormones into the blood, and you have a very close association of the secretory cells with the vascular system. Now we're going to move on to the adrenal gland. And the adrenal gland sits just above the kidney. That is what gives it its name. We're looking at very low magnification at the entire adrenal gland in cross-section. And like many organs within the body that you will encounter, it has two main parts. The cortex, which is labeled C, which I'm tracing with the arrow, and then a central medulla, which is labeled M, which I'm also now tracing with the arrow. The arrow here points to a blood vessel. This is a vein coming out of the medulla, and this is, upon stimulation, the hormones enter the blood and are immediately brought out into the inferior vena cava and then go through the heart and are rapidly spread throughout the body. At higher magnification, we can see these two different parts of the adrenal gland with greater clarity. The C on the outside here, the outermost layer, is the capsule. This is connective tissue. Note that there are blood vessels just beneath the capsule. And then beneath the capsule is the first layer of the cortex, which is called the zona glomerulosa, and that has to do with the appearance of the epithelial-like cells that exist in this outermost portion of the cortex. Below that, or inside that, is the zona fasciculata, labeled F. Note that it stains differently, that is more lightly, than the zona glomerulosa. And then a third layer of the cortex is called the zona reticularis. Again, it stains somewhat differently from the other two layers. And the innermost layer here is an edge of the medulla. At the corners down here, note the very large blood vessels, which facilitate getting hormone out into the blood system very rapidly. And what you will need to understand about the function of the adrenal is that the zona glomerulosa produces a hormone called aldosterone, and that's regulated by hormones secreted from the kidney. The zona fasciculata secretes corticosteroids, and that is regulated by cells in the pituitary, which we saw earlier. Those are the corticotrophs, and they secrete ACTH. The third layer of the cortex, labeled R, the zona reticularis, secrete weak androgens, which you'll learn about in detail not only from Dr. Burdick's, but later in the course as you're learning about the endocrine system in the second semester of physiology. And the zona reticularis secretes androgens, and the medulla secretes two hormones that are related to each other. One is called epinephrine, and the other is called norepinephrine. So what I'd like you to do now that you've had this brief introduction to the endocrine lab is to, as you have done the entire semester, look at all of the images in the endocrine lab and then test yourself on your understanding using the Learn at UW quizzes and study these images together in the histoatlas as you prepare for the quizzes and the exam.